We're very pleased today to have with us, we're doing a site visit at Vanderbilt University into insights into T1D pathogenesis, the Islet perspective. And today we're very excited to welcome Dr. Marcela Brissova, Dr. David Jacobson, Dr. Danielle Dean, Dr. Angela Kruse, and Dr. Jeeyeon Cha. Thank you all very much for joining us. And I just want to give a little bit about each person, just a very short bio. They have much more behind them in terms of their research programs and research expertise and papers published. But just to sort of give you a thumbnail sketch about what they're doing there at Vanderbilt, Marcela Brissova, PhD, is a research professor of medicine, diabetes, endocrinology, and metabolism. She's the director of the Islet Procurement and Analysis Core and the director of Human Islet Phenotyping Program, or HIP. Angela Kruse, PhD, is the research assistant professor of medicine, cell and developmental biology. David Jacobson, PhD, is a professor of medicine, molecular physiology, and biophysics. Jeeyeon Cha, MD, PhD, is an assistant professor of medicine, diabetes, endocrinology, and metabolism, as well as an assistant professor of molecular physiology and biophysics. And the moderator for this talk is Danielle Dean, PhD. She's an assistant professor of diabetes, endocrinology, and metabolism, as well as an assistant professor of molecular physiology and biophysics. Thank you very much. We're looking forward to having Dr. Dean moderate this talk, and I will also take questions that are dropped into the chat. But first of all, I'd like to have Dr. Brissova sort of start us off with her work, and whether or not she'd like to share any slides, it's completely up to her to illustrate her work.
Well, I prepared the slides. I think slides always help. I'm a very visual person, so I may have actually too many slides. So feel
That's
free to
fine.
cut me off at any point you'd like, okay?
We won't. We'll take what you have. We're very interested to see.
All right, all right. Okay. So I thought I would share with you some thoughts on defining molecular signatures of human diabetes by integration of multiple data modalities. And I'm gonna share these thoughts from the perspective of an islet biologist. And I'm gonna be very islet biased, obviously. So here are the forms of diabetes, obviously type 2 being the most prevalent form. Underlying a problem here is the loss of functional beta cell mass, which plays a critical role in all forms of diabetes. Currently, common diagnostic tool is measurement of glucose, and we do not understand molecular or genetic features of islet dysfunction leading to diabetes. My journey to islet biology really began with looking at cytoarchitecture and composition of human and mouse islets. This work was published nearly 20 years ago, and it still is very fresh in my mind. Initial work was supported by Beta Cell Biology Consortium, and then progressed to a human islet research network that is now really focused on human islet biology, and led to development of programs such as HapMap, Human Islet Phenotyping Program of IIDP, and also programs that are supported by private organizations or foundations such as Hemsley Charitable Trust, for which we are very grateful. Why integrative multimodal approaches are important? It's because type 1 and type 2 diabetes are heterogeneous, they are highly polygenic, and it's been very difficult to attribute T1D and T2D genome-wide association study signals to cellular function. You see number of signals for T1D and T2D, and also different stages of type 1 and type 2 diabetes may involve different biological processes. And so what I would like to highlight today is our work that led to development of a roadmap to essentially study human pancreas. So it's focused on early stage type 2 diabetes, but the strength of this approach was really in multimodal data integration and in interdisciplinary approach. And would not be possible without organ donors and their families supporting human islet and pancreas research. So through this multimodal integration, starting really with the tissue imaging and understanding cellular architecture, overlaying physiology and function, adding transcriptomic epigenomic profiling, and all the way to genetic risk variants, we were able to discover a key factor in early stage type 2 diabetes, RFX6 transcription factor as a hub gene. This was work of a really remarkable multidisciplinary team, and this was possible through integrative analysis and data sharing and deposition. I would like to highlight Pancreatlas as a way that was developed here actually at Vanderbilt. Big shout out to my two dear colleagues, Diane Saunders and JP Cartier. It's a platform based on, built on FAIR principles to bring imaging data sets, large multiplex imaging data sets, digital pathology to diabetes research community. And this work would not have been possible without Vanderbilt Pancreas Biorepository and infrastructure that we've built for studying human pancreas and islets that had been adopted by other programs here in the US. I'm very grateful for collaboration with Dr. Rita Bottino, world-renowned expert in islet isolation. And what we've been able to accomplish is to generate tissue and also isolated islets from the same organ donor. And this is all connected to the identified medical record. So Vanderbilt Pancreas Biorepository and Vanderbilt Islet Phenotyping Pipeline were really foundation for the work that had been recently published in Nature. And I would like to highlight our interdisciplinary team across islet biology, statistical genetics, computational resources, transcriptomics, genomics at University of Michigan, dear colleagues, and physician scientists. My mentor and longtime colleague, who was able to identify as board-certified endocrinologists early stage T1D donors for this study published in Nature. And really, what I would like to highlight is that through imaging and multimodal approaches, we were able to show that beta cell loss is not significant in early stage type 2 diabetes. There is normal beta cell, alpha cell, and delta cell mass early on, which has implications for our understanding type 2 diabetes. It is really beta cell dysfunction, loss of glucose-stimulated insulin secretion that underlies a disease process. So it's really intrinsic beta cell defect as a key feature driving reduced insulin secretion in early stage type 2 diabetes. And this integrated approach, this roadmap, can be really applied also to type 1 diabetes. So here at Vanderbilt, we were able to look into alpha cell dysfunction in type 1 diabetes, and also alpha cell dysfunction early on in an autoantibody positive stage prior to symptomatic T1D. And these are efforts that are led by larger programs such as HERN, HPAP, and NPOD. Going forward, I think integration across multiple programs and data sets will provide new diabetes insight across age, gender, sex, across multiple data streams, and connecting really basic science to clinical trials, supporting really interaction of basic scientists and those who are working in the area of clinical and translational research. So I can offer a few reflections before I go. I feel that complex diseases such as diabetes require integrated studies by collaborative interdisciplinary teams. working with human tissue and human islets is very challenging, but it's the path to provide more insights into etiology and pathogenesis of human diabetes. And such approaches should lead to detailed molecular understanding of really heterogeneity across multiple levels within diabetes and provide foundation for new diagnostic tools, disease management, and really leading to prevention. None of this would be possible without organ donors and their families. To me, it's really a gift of life through knowledge that we gain from studying these organs and islets. And as a final highlight, I would like to invite you to an IDDK workshop that is going to be at the end of this month on artificial intelligence in precision medicine for diabetes and other chronic diseases. Here are QR codes for the workshop. It's going to be a hybrid workshop. And there's going to be pre-workshop survey and a couple of pre-workshop webinars that would be fun to join if you are interested. And with that, thank you so much for your attention. And I'm going to pass it to you, Monica.
Thank you so much for that great overview and for all this and for the shout out to the upcoming AI conference. That looks like a really great one. Danielle, did you have any questions?
So I just wanted to ask, so what do you think, what do you think about, what sort of advice would you give to, say, a younger diabetes researcher who is maybe spent a lot of their career focused on the mouse like you were early in your career? And how do you, what kind of advice would you give to them to start to move towards human models, if that's something that they were looking into?
I think the model system that we have are valuable, but it's a model system, right? And with that comes caution in interpretation of the data that we can collect. So whether it's a zebrafish, whether it's mouse, whether it's a human islet or a human pseudo islet or stem cell derived islet-like organoids, I think they are valuable model systems, but we as scientists have to be cautious how we interpret the results and connect them to human disease. I think that would be my advice.
Okay. That's great advice. And so now we'd like to showcase the next researcher in the room, Dr. Angela Kruse.
Great. All right. Well, first of all, thank you so much to the organizers for the opportunity to speak to you today. My name is Angela, and I am part of the Spraggins Lab, as well as the Mass Spec Research Center here at Vanderbilt. And all of the work that I'll show today was done in really close collaboration with the Powers and Brissova labs, and was sort of supported by the NIH Consortium HubMap as well. So I'll jump into it and say that at our Mass Spec Research Center group, we really see disease as a spatial problem, and therefore we want to apply spatial solutions to understand it. I think Marcela did a great job of highlighting that type 1 diabetes is a highly spatial problem. And one thing we're quite interested in is what are the molecular differences in islets that are next door neighbors, and one might be performing quite well and the other one has a really low response. So with that in mind, we're developing a kind of what we see as a bioanalytical toolbox to understand spatial problems. And that includes the integration of technologies such as sequencing and spatial transcriptomics with other technologies such as microscopy, for example, tagged microscopy like Codex, along with stained microscopy and even in vivo imaging. But really at the center of our toolbox is imaging mass spectrometry. And I'll kind of show how that works and how we're focused on implementing it to study diabetes and integrating it with some of these other technologies. Okay, so imaging mass spectrometry, in brief, starts with a tissue sample that has been cryosectioned onto a glass slide. And then matrix, which is a small molecule matrix, is applied to the tissue surface, and this molecule helps to extract and ionize analytes on the tissue's surface. Now, these analytes can be anything from lipids to small metabolites or even intact proteins or glycans. Now, the data that I'll show you today is going to be on lipids, but we'll get to that. So, all right, we have a tissue section on a slide. We cover it with a homogeneous coating of matrix, and then we put it into a mass spectrometer, and we do MALDI, matrix-assisted laser desorption ionization. And what that means is we shoot our tissue sample with a UV laser, and each time the laser hits the surface, it generates a plume of ions that we can measure. We do this in a raster or grid-like pattern across our sample, and each laser shot generates a spectrum measuring hundreds or thousands of ions. And then we can reconstruct these spectra computationally, where each one becomes a pixel. And then we can reconstruct those into an ion image that shows us the relative abundance and localization of the ion that we measured. All right, so what does that look like for the pancreas? Now, I realize this is kind of a lot on a slide, but I'm just going to walk from the top left here. So in the case of the pancreas, we've taken human donor samples, and we've performed imaging mass spectrometry looking at lipids in our samples. And we're also able to do immunofluorescence to identify abundant cell types in these tissue samples. So on the left, I'm showing an antibody stain where beta cells are green using an antibody for insulin, and the alpha cells are shown in purple. And then on the right, what's quite exciting is that we can do our imaging mass spectrometry experiment and really uncover the lipid landscape of these samples. And one thing that's quite important is that this is an untargeted assay, so we don't need to know what lipids we are looking for. We're discovering what's there. So furthermore, we can look at the kind of lipid changes in patient samples from individuals that are autoantibody positive, shown here, compared to those who have been diagnosed with type 1 diabetes. And I just want to highlight one thing here in the middle. If we look at this cluster of islets, again, these alpha cells are marked in purple. And then we look at the lipid distribution on the right. This is just showing 4 lipids that we identified out of hundreds. Hopefully you can visually see that this lipid in yellow certainly seems to be localizing to these alpha cells, but not the beta cells. And furthermore, this lipid in red really seems to be more abundant in these beta cells. And this in and of itself is actually quite exciting because 10 years ago, we did not have the technology and spatial resolution to distinguish these cell types, which of course are approximately 10 microns in size. But even more exciting than that, we can annotate these islets and look at differences in the relative abundance of these lipids across patient islet groups. So for example, this lipid in yellow is much more high in abundance in these islets from a type 1 diabetic sample compared to non-diabetic and autoantibody positive. And what we're quite interested in as well is these lipids that seem to have a gradual increase as we move from non-diabetic through autoantibody positive and then to fully type 1 diabetic. So we think that this will help us uncover potential lipid biomarkers that might be accumulating during the development of disease. So this is kind of our core technology. And then I want to close with talking about how we're integrating it with other molecular imaging technologies as well. So here I'm showing imaging mass spectrometry for lipids on the right. That's what we've just been looking at. And then we've actually now been able to also perform spatial transcriptomics, in this case with Xenium. This is a 10x technology. And this allows us to actually cluster the cells in our sample according to their transcriptional profiles. And then we can, on the same section, perform codex multiplexed immunofluorescence. Now, this is looking at protein targets. And each of these technologies really brings something unique to the table. So for example, Codex helps us annotate these cell types at really high spatial resolution, and then also establish the ratio of cell type in each islet. The spatial transcriptomics allows us to kind of separate and see if there are groups of islet cells that share unique transcriptional profiles that might differ from each other. And then finally, this imaging mass spectrometry data is untargeted kind of discovery of the lipid landscape. So once we integrate these technologies, we can do something like what I'm showing on the left, which is just one single islet where we've co-registered Codex and Xenium data. And then what we can do is segment those cells so that we can have masks for something like alpha cells compared to beta cells in this islet. And once we have those locations, we can then retrieve the average lipid spectrum for these major cell types. So that's on the right of the slide. This is a different spectrum comparing the lipid sort of signatures for alpha cells compared to beta cells. And you can see here that there are several lipids which have enormous variations in intensity. And then finally, what we can do is apply this across all of the major cell types that we can identify with Codex and Xenium. And this is just showing 3 examples on the left. This is the mask showing the location of alpha cells compared to beta cells, compared to mast cells in our sample. And then here we can look at the variation in lipid intensity across these major cell types. So I'm just showing one example here. This is a phosphatidylserine lipid, and you can see that it has the highest abundance in beta cells compared to alpha. And so we can start to uncover the kind of lipid barcode of each of these cell types, and then uncover which of these are accumulating during the development of disease, and then how that varies across patient demographics and gender. So finally, I just want to say that this work is highly collaborative with our university as well as others, and was supported by the NIH HubMap. And then finally, I want to thank the donors and donor families without whom studies like this with human tissue would not be possible. And I'd be happy to take any questions.
Thank you very much, Angela. Danielle, do you have any questions for Angela?
I do. Actually, I think this is fascinating work. So my question is, you mentioned some of the lipids that were unidentified, and then even who had done a lot of the lipid identification. Help us understand the bottleneck, or the enormous magnitude of the amount of data you get, and how you think that moving forward, you might address identifying these things.
Yeah, that's a super important question. So with imaging mass spectrometry, oftentimes we just get, it's kind of like a scale, right? So oftentimes we just get the mass to charge ratio of a molecule, and then we have to identify what it is. And that can be an enormous challenge because sometimes molecules don't fragment very easily, such as large proteins are really difficult to fragment. So what we've been doing, and I really want to highlight the work of Dr. Madeleine Colley, who's a postdoc in our group. What we've been doing with her is she will do a LC-MS lipidomics of every single patient sample that we get, and build a custom library of known identified lipids. And then we match that with our imaging mass spectrometry data. So that is kind of a bottleneck in that she really had to do a lot of work to nail down those methods. And then the bottleneck can be computational in that we have to match up all of those masses. So we also have a great collaboration with Ralph Bondaplas's group, and they just released a new software called Anatine that does those matches. So I think that we're starting to really address some of those things, but there's exciting work to be done.
I think we have a question in the chat from
great.
Yes, we
Ian.
do. From Ian Sweet, the descriptive power of the technique is amazing, but how do we move from, say, lipids to mechanistically what is going on during and after the development of the disease? Thanks so much.
Oh yeah, I think that's an exciting question as well, and I'm open to discussion on it. All right, so yes, we've made enormous strides in kind of uncovering where these lipids are in the case of disease. I think that what we're now doing is kind of overlaying that with more knowns, I would say, so like ground truth studies. So for example, we're starting to couple our lipid imaging with apoptotic stains so that we can identify some lipids like phosphatidylserines, which are kind of have been associated with apoptosis. But if we can really nail down what species, for example, are present in apoptotic cells versus cells in other states, I think as we start to combine our lipid imaging with other kind of longer existent technologies, that can really help us. And furthermore, I think there's really exciting work done on lipid rafts and how lipids kind of assist proteins from talking to each other and cells from talking to each other. And so I think there's some interesting work to be done there, whether we can uncover, the lipid profile and then connect that directly with proteins, which I think is becoming more possible now with the integration of Codex.
Yeah, I guess can I follow up here?
Yes, thanks, Ian.
I mean, do you need, more information to know, what the situation was before, symptoms or before, right? This is such a hard thing to study when you have such a long period of time that the disease develops. And so, do you have an idea of how you can distinguish between, causality and correlation and what's, what are the mechanisms that we need to block to prevent the disease? That's
Yeah, I think
Very
that's
daunting, right?
definitely daunting. And I think the way that we're thinking about it right now, and open to your thoughts as well, is that we're generating these kind of hypothesis generating resources. And I think that where it becomes more powerful is where we can partner with biologists like Danielle, who can kind of specifically test these mechanistic questions. And I think that's kind of where we start to uncover the mechanism, is testable hypotheses that we can then confirm with orthogonal studies in mice and maybe even pseudo islets and cell culture lines.
Right. Yeah. No, very heroic. Keep up the good
That
work.
was a great question, Ian. And I mean, the power of our work related to early stage type 2 diabetes was to go after disease driving mechanisms rather than consequences of the disease, right? That was so important. And, you know, the fact that SNPs in non-coding part of the sequence influence RFX6 expression and are causal to T2D. That was very important.
Right.
That would not have been possible without the multiple streams of data and data integration, really vertical integration.
Yeah, no, I'm going to give a lot of thought of how I can use this data and to correlate the things that I'm looking at, right? That, for instance, beta cell death from antibodies, you know, how could this be related to the lipids and the different formations? In fact, we're going to be studying that with a rat model, which isn't obviously the human, but if we see some of these some progressions of cell death in our models, I mean, is this something you guys can then measure in rodents? Would we send islets? How would that work?
Yeah, I would say, to jump in, our technologies are highly compatible with rodent systems and model systems. So I think that would be amazing. I think that that's where we'd ultimately love to get to, is generating these hypotheses in very precious human tissue and then testing and confirming in tractable systems like rodents.
Yeah.
So yeah,
Good.
shoot us an email.
I
Great.
will.
A spark of a collaboration. Here's a question from Kiati Gerder in Boston. Could you use the same technique for the small metabolite?
Yes, definitely. So we have a really talented postdoc, I'll highlight, Ali Zahadi. So he's been doing beautiful work on small metabolite imaging. He's been doing it in the human eye, but his methods would be, I think, a perfect application in the human pancreas. I would just add there are some technical considerations. So with small metabolites, Danielle can comment on this as well. We just need to be really careful to image those things really quickly, because metabolites are quite susceptible to oxidation. And then we also we have to minimize any washing of the tissue, because they're also sometimes quite hydrophilic and can delocalize. So we just have to be quite cautious with how we treat our samples, but we have great workflows, and I think that would be a good idea.
Fantastic. Any more questions on this session? All right. We did ask a question sort of off the record, and I did wonder if you could just comment briefly on, you know, the microbiome has shown to be changed during the course of type 1 diabetes, during the stages and the presentation of type 1 diabetes. Some of those microbial bacteria, some of the bacteria in the gut are producing lipids, and you wondered, how is your system have a way to track lipids produced in the gut versus, you know, lipids that are inherent to the islets?
Yeah, I thought that was a really exciting question. So the one thing that we the way that we're thinking about it is to couple like fluorescent in situ hybridization, which can be specific for bacterial species, with imaging mass spectrometry. So we've had success in doing that in some other organ systems. And so I think that would be really exciting to apply in the pancreas. And then the other thing I would add is that bacteria tend to produce odd chain lipids. So they have an odd number of carbons. And I think that would be a really exciting way to kind of parse the data, looking at odd chain lipids compared to even chains, which are usually produced by mammalian cells. So I think that's an exciting area of future work.
Great, thank you so much. Yeah, it's very interesting because there is a big decrease in microbial diversity at the time of diagnosis. So it might be interesting to see what's happening using your systems and pipelines. Okay, great. We're going on to the next presenter, Dr. David Jacobson.
So I'm going to tell you a different focus. Our focus is on ion channels, and I'm going to tell you why in just a second. Let me get my pointer going here. So we're interested in how ion channels modulate islet function and health. And the reason for this comes from some early work by Gerald Grodsky back in the 60s, where he looked at ions in solution to see which ones were important for beta cell function. And so what he did is he perfused a pancreas with high glucose, with or without calcium. And here's where calcium was present in the media. And I think you can appreciate right here, these closed circles. Insulin secretion doesn't occur unless you have calcium in the media. And so this was the first evidence that calcium entry into beta cells is required for insulin secretion. And it was subsequently shown that the calcium gets into the beta cell through these voltage-dependent calcium channels, leading to granule fusion with the membrane and release of insulin. And so we're curious, because it's so important, how calcium entry is regulated. And so the canonical model of calcium handling in the beta cell is that glucose gets into the beta cell through glucose transporters, is metabolized, it leads to closure of a potassium channel that's ATP sensitive. This is termed the KATP channel. Most people know about this. Mutations cause neonatal diabetes. And when this channel closes, this leads to membrane depolarization, allowing activation of the channels I just mentioned, the voltage-dependent calcium channels. Calcium gets in and leads to insulin secretion. And while this is really important, there's numerous other channels that are critical to beta cell function, and I'm going to tell you about one of those today. So another reason that we're interested in calcium handling is not only based on secretion, which is glucose dependent, but on the numerous signaling pathways. This is just a few of them in the beta cell that are sensitive to calcium. And so mitochondrial function is dependent on calcium, ATP synthesis, and reactive oxygen species generation. The endoplasmic reticulum is a calcium store within the beta cell and is critical for insulin production, processing, as well as a stress response which occurs when the cells, such as in type 1 and type 2 diabetes, are overworked, and this helps protect the beta cells from death or destruction. And then also in the nucleus, calcium is critical for transcription, chromatin remodeling, and many of the gene networks that are prevalent in the beta cell are dependent on various transcription factors that are calcium sensitive. And so it's important to study calcium and for many reasons in the beta cell, not just secretion. So what happens in type 1 and type 2 diabetes is that the autoimmune attack, as everybody is well aware, leads to beta cell destruction. It also right away causes changes in beta cell calcium handling. But once beta cells become overworked in both type 1 and type 2 diabetes, you have excess beta cell calcium influx. And this eventually leads to beta cell stress and eventual dysfunction and death. Following the overworked beta cell, your endoplasmic reticulum has to produce a ton of insulin and begins to become stressed. And under these conditions, eventually endoplasmic reticulum calcium stores are diminished, and this leads to further stress and deterioration of the beta cell through oxidative damage and eventual apoptosis. And so it's really critical to understand calcium handling because they could be targeted to prevent beta cell dysfunction during the pathogenesis of diabetes. So it's important to note that calcium handling has been targeted in type 1 diabetes. And this was some work from Aneth Shalev's. This is just one example. Aneth Shalev's group in 2018, where they treated type 1 diabetics that were recent onset type 1 diabetics with a calcium channel inhibitor, verapamil. And the reason they did this is that they knew when the beta cells are overworked, there's excess calcium, and this leads to stress and diminished beta cell function. And interestingly, when they treated these patients with verapamil, this caused a restoration of beta cell function, leading to increased insulin secretion under glucose-stimulated conditions. And it took the stress off of the beta cell. And so targeting calcium can be used as a treatment, not only for type 1, but also for type 2 diabetes. As I mentioned, the endoplasmic reticulum also serves a critical role in calcium handling. And so we're also interested not only in how ion channels modulate plasma membrane calcium handling and calcium entry, but also how it modulates endoplasmic reticulum calcium homeostasis. And so under physiological conditions, the ER calcium is high, and calcium release from the ER stimulates insulin secretion, is transferred to mitochondria to stimulate mitochondrial function and proper ATP production, as well as stimulating chaperone function and protein folding and proper protein production, such as insulin. Under diabetogenic stress, such as type 1 diabetes, calcium eventually becomes depleted in the ER. This leads to decreased insulin secretion, translation inhibition, reducing protein production, severe mitochondrial dysfunction, and protein misfolding. And so this really contributes to beta cell destruction during the late onset or the late stages of type 1 diabetes. So beta cell calcium influx and ER calcium release are controlled by potassium channels. And I'm going to tell you about a few of these. The ones that one of the examples of a channel that we work on are the 2 pore domain potassium channels. TOC1 and TOC2 are the ones that we're currently working on. And these are found both at the plasma membrane as well as the ER membrane. And movement of potassium on both membranes leads to loss or gain of negative charge on the inside of the membrane. On the ER, it diminishes negative charge, and on the plasma membrane, it builds up negative charge at the membrane. So at the plasma membrane, potassium channels inhibit voltage-dependent calcium channels, while on the ER membrane, they're turned around and they actually stimulate ER calcium release by allowing for an electrochemical driving force to be maintained. So TOC1 is the most abundant potassium channel of the human beta cells and shows islet restricted expression. And so casein K16 is the gene encoding TOC1, this 2 pore domain potassium channel. And you can see in human tissues that it's only found in the pancreas. And when we stain for TOC1, what we find is it's only found in endocrine cells within the pancreas. And so this is great for a target for diabetes because you don't have expression in the heart, the brain, or other parts that could be affected by drugs that target this channel. Importantly, if you look at TOC1, it's not just at the membrane. As I mentioned, it's also intracellular, and it co-localizes with ER localized markers, in this case calreticulin. If you overexpress it with an ER tagged marker, you can see it co-localizes quite nicely with that ER marker. And when we record ER membrane currents, TOC1 can be found as functional channels on the ER membrane. And so we wanted to see how TOC1 modulates endoplasmic reticulum calcium handling. And to do this, we used this genetically encoded calcium sensor, which is targeted to the endoplasmic reticulum. And then we looked in islets from animals either with or without TOC1 channels. And I think you can appreciate here that the calcium within the ER was much higher when you lose TOC1 channels in the endoplasmic reticulum. And this really indicates that TOC1 diminishes ER calcium by increasing the driving force for calcium release from the endoplasmic reticulum. And this occurs at both low and high glucose conditions. And so what we've determined over the course of these studies, and I don't have time to go into all the details, is that TOC1 channel potassium flux mediates a countercurrent for more efficient endoplasmic reticulum calcium release. And so the question became, especially for type 1 diabetes, can TOC1 inhibitors be used to selectively elevate beta cell endoplasmic reticulum calcium storage and prevent beta cell ER stress and destruction? And so to start looking at this, we had to develop pharmacology for TOC1 channels. And to do this, we generated a cell line with conditional overexpression of TOC1 channels with tetracycline inducible TOC1. You can see protein for TOC1, you get nice induction with tetracycline. We can plate these cells on a 384-volt plate and treat them with compounds. We screened 50,000 compounds from the Vanderbilt Chemical Library, and we had to generate a fluorescent assay for TOC1 channel function. And this was actually designed by David Weber when he was here at Vanderbilt. It's called a thallium fluorescent assay. And what he's done or realized is that thallium is the same radius as potassium, and so it can get through potassium channels. And you can load cells with a dye for thallium. And so when you add thallium to the outside of the cell, such as here, you get this influx of thallium that you can read when the thallium binds to that dye. And when we induce TOC1, you can really appreciate that there's a big increase in that thallium influx. And so that's a nice readout for TOC1, and we can do this really rapidly. And so like I mentioned, we screened 50,000 small molecules, and we recovered 187 TOC1 inhibitors. We've since screened against many other channels, including the closest relative, TOC2, and we've now got 50 inhibitors that we're currently investigating. These are some of the inhibitors. We have IC50s in the low micromolar level. And so it's not perfect yet. We're developing or making them more potent for medicine currently. But we wanted to see if they would work to prevent beta cell dysfunction. And so we've started doing this. One of the ways we've looked at this is looking at ER calcium storage. And so when we induce overexpression of TOC1, what you can appreciate here, this is uninduced here, and this is induced here. It drops ER calcium storage significantly. And that's measured by just causing all the calcium to be leaked out of the endoplasmic reticulum by blocking the sarcoplasmic endoplasmic reticulum calcium ATPase, which is a pump for calcium entry into the ER. And I think you can appreciate when you do this, in cells expressing TOC1, they show the lowest amount of ER calcium release. And when we treat these cells with an inhibitor to TOC1, that's increased significantly. And so it indicates that blocking TOC1 can be used to selectively elevate ER calcium storage. And the nice part about these inhibitors is they will only block or increase ER calcium in the islet and not in heart muscle or in neurons or any other tissue, which is good. And so this is just one example of an ion channel study that we're investigating right now. We have many others on other calcium handling proteins, including pumps, but I don't have enough time to go into those. And so what we're trying to do is go from target to drug and seeing if any of these can be used to treat type 1 or type 2 diabetes. So with that, I need to thank all the people that did the work that I showed. This was work done by Arya, Sarah, Prasanna, and some of the new graduate students. They've since left the lab. And I need to thank the funding, NIDDK, ADA, JDRF, Breakthrough Type 1, the Helmsley Charitable Trust, and Vanderbilt, and the donors for which this human islet work that we've done would not be possible. And so I'd be happy to answer any questions. I know you have one, Ian.
I do. I was typing it, but maybe I'll just say it. Well, so this is great stuff. I mean, just what I like to see. Brodsky, no question, got it right. But I was puzzled at some point in some of the data that I've gotten with thapsigargan, which shows an increase in cytosolic calcium when you give, presumably because less ER calcium's coming out, but no change in insulin secretion. And so I was always wondering what the evidence for an actual relationship between calcium release from the ER and increased insulin secretion. And there's no question the L-type channel is the most important one, but what's your view on that?
So yeah, it's a great question. So thapsigargan blocks SERCA, which is this pump that pumps calcium into the ER. And when you do this, you release calcium. And currently, one of the things we really need to figure out is how calcium leaks out of the ER. It's really important for setting baseline calcium, but that leak process is quite slow. If you stimulate a beta cell with a G protein coupled receptor ligand, a GQ ligand, such as acetylcholine, now you stimulate these receptors, such as IP3 receptors, which are calcium channels, and allow much more rapid calcium release from the ER. In fact, ryanodine receptors are even faster than the IP3 receptors. And so I think it has to do with the speed of calcium release, and that GQ and ryanodine coupled release of calcium from the ER can definitely stimulate secretion. But I think it's a great question, and we really need to start understanding what the calcium leak channels are, because these are going to be really important to setting not only the ER stress response, but also basal cytoplasmic calcium levels.
So just to follow, we should have a beer over this, because I followed up when I saw that thapsigargan data, and we actually gave acetylcholine to islets, and we did it before and after giving thapsigargan. And so, obviously acetylcholine is very potent in increasing insulin secretion, but if you give thapsigargan first, empty the ER out, acetylcholine then has no calcium response, but still has an insulin secretory response. And I interpret that being that it's protein kinase C that's really the mediator of acetylcholine response. And again, it's sort of consistent with maybe my biased view, but that ER calcium probably has slow effects and definitely can affect ER stress and a lot of things you were talking about. But as far as the acute stimulation of insulin secretion, I guess I'm still waiting for more definitive data to see that it takes a role like the L-type channels do.
Yeah, and I agree that the ER response is much different in terms of secretory response than, say, the L-type calcium channels, which are allowing massive calcium influx right by the granules. But there is some evidence that the ER contacts the plasma membrane, and this is close to granule pools. That could be really important, not only with store-operated calcium entry, which is mediated by ER calcium depletion and causing some secretion, but also in second phase insulin secretion, where the granules are coming up to the membrane and being secreted. And interestingly, when you treat islets with dapagliflozin and or CPA, what you see is a significant change in the calcium waves that are responsible for pulsatile insulin secretion. They become flat, and so they don't have this nice curve down, and they're much faster. And so the calcium release from the ER is critical, not only in regulating secretion, but also regulating ion channels at the membrane, one of which is termed K-slow, that regulates the silent phase between oscillations, which is critical for pulsatile insulin secretion, which becomes defective in type 1 and type 2 diabetes, leading to changes in insulin sensitivity. And so I do think there is some link between ER calcium as not only release, but also signaling to control calcium handling and secretion. And I think it needs to be worked out. I think there are some people that think it's all ER calcium. Marjan Rupnik and others think it's IP3 receptors and ryanodine receptors that are solely responsible for glucose-stimulated insulin secretion. And one of the things he mentioned, which I think is quite interesting, is if you starve your cells for too long, you actually deplete ER calcium stores, which makes sense. And the reason for this is the plasma membrane calcium ATPase has a much higher affinity than SERCA for calcium. And so as calcium leaks out of the ER, it's pumped out of the cell. And so with low glucose, where you don't have any calcium coming in, eventually the ER becomes depleted because you don't have enough ATP to energize SERCA, and you have dumping of ER calcium. And so I think one of the things we have to do as a team is to use glucose more euglycemic levels instead of starving them at 2 or 1 before we do our experiments.
Yeah, good point. Thanks.
Yep.
That was a great back and forth discussion, really fascinating. Thank you both for participating there. I'm just going to say, in the interest of time, I'm going to go directly to Jeeyeon Cha, MD, PhD, and she's going to discuss her work. Oh, I guess before you leave, can you just answer this, David? Are there any hypotheses around why TOC1 is only present in beta cells?
Yeah, well, I mean, one of the big things about the beta cell that, oops, I'm sorry, I can't get my, the beta cell has to handle a lot of protein. And so what we found is there are certain 2-portoamine potassium channels that are really important for ER calcium handling. And 2 of those are TOC1 and TOC2. Both have SNPs associated with type 2 diabetes. And TOC1 actually has a mutation that causes maturity onset diabetes of the young. And what we think is that the ER calcium is so critical for insulin production, as well as the ER stress response to prevent beta cells from dying, that it has to be a high level of these channels to modulate the function within the beta cell. And that's our hypothesis right now. We would love to see if there was a ligand specifically in the beta cell that was responsible for controlling these. One of those ligands that's recently been identified is long-chain acetyl-CoAs, and these are really important during glucose stimulation as well as under type 2 conditions where they go very high. And so it could be that that's an important signal for tuning the beta cell and preventing from diabetic phenotypes. One of the things that happens with gain of functions is that you develop diabetes earlier, and so we think this is dumping ER calcium, but we're not exactly sure, and a lot of work is needed to sort that question. It's a great question.
Great, thank you. Just that we're hitting sort of the high notes here and the tips of the iceberg to really whet people's appetite to learn more and read more of the papers coming from these excellent laboratories. So thanks for bearing with us as we have these sort of vignettes. And Jeeyeon, would you like to take over now?
Thank you. Those are very hard acts to follow with the beautiful studies. But I will just introduce my lab and my work. So I'm Jeeyeon Cha. I just opened my own shop here at Vanderbilt in late 2022. So my story is probably not as developed, but we have some really exciting directions. So our lab studies the molecular mechanisms underlying pancreatic islet dysfunction and diabetes, but also particularly from the lens of biologic sex.
Now, we know that there is a increased signal for type 2 diabetes in men over women, although the degree of this discrepancy does vary by population. And unusually, in type 1 diabetes, there seems to be an equal prevalence across males and females, although it is an autoimmune condition, which typically falls on females. So there seems to be some sex bias in terms of vulnerability for beta cell and islet dysfunction. Now, our lab studies a model of sex-dependent diabetes resulting from a mutation in an islet-enriched transcription factor called MAF-A. And interestingly enough, the models that we're studying that produce diabetes or exhibit diabetes have accelerated islet aging and cellular senescence, which is a common feature with type 1 and type 2 diabetes, both in human studies and in mouse models. So what do we know about cellular aging in the beta cell? So senescence and cellular aging is a durable cell cycle arrest that's associated with aging and aging-related diseases. Healthy beta cells also exhibit a physiologic senescence associated with increased age and functional maturity. However, pathologically accelerated beta cell senescence is seen in human diabetes at rates of even 50 to 60% of the beta cell population in type 2 diabetes. And intriguingly, in mouse models of type 1 and type 2 diabetes, senescent beta cells are seen, and the removal of them you see a recovery of insulin secretion and improved beta cell function, suggesting that these senescent beta cell populations are indeed pathogenic. Now, senescence is not a rigid molecular entity. The molecular signatures are actually variable depending on the etiology. So in this nice schematic that I've adapted from our collaborator, Peter Thompson at the University of Manitoba, in aging-related senescence, this is typically characterized by an induction of P16, P19 cell cycle inhibitors as the beta cell functionally matures. However, in pathologically accelerated senescence, which is typically associated with a DNA damage or some sort of cellular stress or damage response, this induces cell cycle inhibition early on with the activation of P21 or P16, and elicits a DNA damage response. So gamma H2AX, 53BP1, typical DNA damage markers you would see are activated early on. Now, to even add more complexity to senescence, there is a temporal progression associated with this pathology. Now, later on, the cells adopt a pro-survival phenotype. They start to express factors in the BCL2 family, which are pro-survival anti-apoptotic factors. And then terminally, they develop what's called a senescence-associated secretory phenotype, or SASP. This is a functional phenotype where they secrete a repertoire of molecules, including cytokines, matrix metalloproteases, growth factors, hormones, et cetera, to induce a paracrine senescence in neighboring cells. Now, this repertoire also changes depending on the etiology and the cell context. However, it seems to be one of the key features of identifying senescent cells. So just to give you to characterize the mouse model that we are using, this mutant MAF-A mouse shows a sex-dependent diabetes. Now, here are some kind of classic glucose tolerance tests in males and females, red being the results of male mutant mice compared to their wild-type counterparts at 4 weeks of age, and the teal being the female hets compared to their wild-type counterparts. As you can see, at 4 weeks, they're similarly showing improved glucose tolerance. But by 5 weeks, there's a dramatic shift in the male phenotype in which they show impaired glucose tolerance, and they maintain this phenotype for the rest of adulthood. Now, this phenotype is associated with accelerated beta cell senescence and islet senescence. This is characterized by shown here by senescence-associated beta-galactosidase staining, which is a kind of a common screening tool for identifying senescent cells. As you can see, at 7 weeks of age, the mutant male islets show increased beta-gal staining comparable to islets from an aged 12-month-old mouse. And this is in stark contrast to female mutant mice, which don't show impaired glucose tolerance and don't show this enrichment of senescence beta-gal. Now just to describe some of the future directions that my lab is taking, we really want to further characterize beta cell senescence. It has such wide-ranging implications in multiple forms of diabetes. So we want to better characterize these senescent beta cells in terms of their secretory repertoire. We hope to pursue mass spec analysis to identify the secreted SAS constituents. We have a panel that we have developed, however, going in a more unbiased manner would be a greater service. And then inhibition of SASP and senescence mediator using known senolytics. We hope to target this pro-survival phenotype, BCL2 family members, and also BET family of coactivators, which has been implicated in developing the SAS phenotypes in beta cells. And then a more long-term project is to really understand what is driving these sex-dependent phenotypes in the presence of this mutant MAFA, such that only the males seem to be vulnerable to develop beta cell senescence. And with the great resources we have at Vanderbilt, we hope to pursue a multi-omics analysis in both mouse and human islets to identify unique beta cell populations that could be driving these phenotypes.
Thank you so much, Jeeyeon. I appreciate that you got such a short time. And we will actually revisit you at a longer talk in the future, just because this is such an important topic and we really want to explore it further. We have 3 others coming next, and we're at the top of the hour. So I apologize, about our timing, but your work is really beautiful and really interesting. And so we would love to showcase it at a longer time in the future. So thank you again for our speakers. And now we're going to shift gears for a little bit and talk a bit about insights into T1D pathogenesis, the immune perspective. So we're going to, for those of you who have, just been here for the Islet part, that's fine. You're, free to go on about your days and business. And for those who are joining for the immune perspective, we have Dr. Dan Moore, Dr. Christopher Wilson, and moderator Al Powers, and we're going to set them up right now.